Hey, good morning, everyone. I'm Ari Sobando. I'm the Assistant Dean for Research and Advanced Studies of the Gokongwei College of Engineering, and I will be your host for today's lecture series. And together with me, we have Dr. Kathleen Aviso, the Dean of the Gokongwei College of Engineering of the De La Salle University. Also, we have Dr. Raymond Tan, the Vice President for Research and Innovation of De La Salle University. Together with us, we have Mr. Rodel Masilungan from the University Sustainability Office. So let me just share my screen. So our lecture today is entitled Decoding Diet with Genomics. And we are blessed to have our speaker. But before we start officially the lecture for today, let us have a short prayer.
Let me be the change I want to see, to do with strength and wisdom all that needs to be done, and become the hope that I can be. Set me free from my fears and hesitation. Grant me courage and fill me with spirit to face the challenge and start the change I long to see. Today I start the change I want to see. Even if I'm not the light, I can be the spark in faith, service, and communion. Let us start the change we want to see, the change that begins in me. Today I start the change I want to see.
Even if I'm not the light,
Even if I'm not the light, I can be the spark. Faith, service, and communion. Let us start the change we want to see, the change that begins in me.
Jesus in us.
Jesus in
Okay, again, good morning, everyone. Good afternoon and good evening to our participants across the globe. I would like to acknowledge the presence of our Chancellor Emeritus at De La Salle University, Dr. Carmelita Quebenco. Good morning, ma'am. And now to give an overview of this LaSalle Sustainability Lecture Series, we have the Vice President for Research and Innovation of De La Salle University, Manila, Philippines. Let us all welcome Dr. Raymond Tan.
Thank you, Aris. Good day, everyone. It's my pleasure to welcome you to this edition of the Sustainability Lecture Series. This is a series of talks that has been co-hosted by DLSU with the International Association of LaSalle Universities, or IALU, since January of 2021. IALU is a global network of Lasallian higher education institution, which has presence in North and South America, in Europe, Africa, and Asia. And the initial intention of the Sustainability Lecture Series was to provide a platform for scholars from the member institutions to share their work both within the network and to friends and colleagues from outside of the network as well. We've held the lecture series in various formats and various languages. Some of them have been held face-to-face, and the series is going strong well into its fourth year. This month, De La Salle University begins its year-long celebration of its Golden Jubilee year. It was in February 1975 that we received our university charter and thus expanded the scope of our mission to knowledge generation and research. And we believe that this lecture series is an important component of how DLSU, in its aspiration to become a globally influential research university, is able to mainstream the work of specialists to reach a much broader audience. Finally, let me just thank the core team, Chancellor Emeritus Skibenko, Professor Aviso, Professor Ubando, Dr. Ria Migo Sumagam, our former PhD student, and Professor Alvin Colaba, who have, of course, kept this lecture series going. And starting this year in partnership with the University Sustainability Office, led by Executive Director Maralit. Most of all, of course, I want to thank our Balik scientist, Dr. Lawrence David, for sharing his expertise on something I personally am keenly interested in, diet and how it affects our health. So I wish everybody a fruitful and engaging talk over the next 60 minutes. Thank you and good day.
Thank you so much, Dr. Raymond Tan. Allow me to share my screen again. All right, so we are now ready for the lecture, which is entitled Decoding Diet with Genomics. And to introduce our speaker, Dr. Lawrence David is an associate professor in Duke University's Department of Molecular Genetics and Microbiology and associate director of the Duke Microbiome Center. The David Lab studies the relationships between diet, the gut microbiome, and human health. The lab is also interested in engineering new tools at the interface of nutrition and microbiology, including building genomic approaches for tracking food intake and microfluidic techniques for high-throughput assay of microbial metabolism. Prior to Duke, Dr. David was a junior fellow at the Harvard Society of Fellows, and he received a PhD in computational systems biology from the Massachusetts Institute of Technology and a Bachelor of Science degree in biomedical engineering from Columbia University. Dr. David has been named one of the 10 scientists under 40 years old to watch by Science News. He is a multi-awarded innovator and investigator, and among his awards are the Burroughs Wellcome Fund, the Balik Scientist, Searle Schmidt Polymaths and Fulbright Scholars Program, the Arnold and Mabel Beckman, Hartwell, Alfred P. Sloan, Damon Runyon, and the Chan Zuckerberg Foundation. Let's all welcome our speaker for this morning, Dr. Lawrence David.
Thank you so much for Dr. Tan and Dr. Aviso for the warm invitation to speak. It's really an honor to speak in such an august lecture series. And see. So yeah, just I'm excited to speak about my work. Oops, wow, it's just starting to go by itself. I didn't want that. Let me see if I can turn that off. Okay, hopefully it will stop scrolling on its own. And so, yeah, as your introduction mentioned, I'm happen to be a Blick scientist. And in case you're curious, I'm a Filipino American, albeit I'm very grateful to be hosted by De La Salle University for this year, as well as the Department of Science and Technology's Food and Nutrition Research Institute in Taguig. I'm on sabbatical from Duke for this 2024 and 2025 academic year, and I'm extremely excited to be with you all. And I especially wanted to thank Dr. Angie Lau, who's been sponsoring me at La Salle for the past couple of months. So I should also mention that I'll be talking about food and nutrition and genomics today. But I wanted to acknowledge that I feel a little guilty. I'm not trained in sustainability. Our lab doesn't work on sustainability outright just yet. My background is in ecology and genomics and microbiology, which are topics that I think are very adjacent to sustainability. And so in fact, I'm very excited to give this talk today. But I also wanted to acknowledge that this is a little bit outside of what I normally talk about. And instead, what our lab primarily works on is these days is nutrition and food and the genomics of it and how we track it. And so I wanted to start by kind of giving you a picture on the left of the kinds of things our lab does. What we do is catalog the genetic diversity of things that humans eat around the world. And so on the left, for example, one of our projects was to go to a local supermarket back in the States, buy one of everything in the supermarket, take pictures, take little bits of samples for genetic analysis, and then we eat the rest. So I feel like I have one of the greatest jobs on the planet, which is I just get to eat all the time. Okay, so why do we study what we do, and how is it relevant to our world? So first, our lab works on nutrition because I'm increasingly convinced that it's probably one of the great health questions of today. So unfortunately, chronic diseases are often linked to challenges with nutrition. This is right now true in kind of economies like in the US, and also I think increasingly true around the world, including the global South, where chronic diseases are overtaking infectious ones as being the leading drivers of reducing mortality and increasing morbidity. So in the US, but again, this is true, I think, increasingly around the world, poor diet is the leading modifiable risk factor for early mortality. So this epidemiological data collected by I believe epidemiologists in the US, and it astonished me to see that poor diet now is considered a riskier behavior than smoking, than alcohol and drug use, than unsafe sex, and even a higher risk factor than things like high blood pressure or high blood glucose. I think what's relevant also to what we're talking about today, though, and I think something that often gets kind of, people often think about nutrition in sort of like a silo by itself, but also for when you think about sustainability, nutrition shapes so much of our environment. So, half the land on the planet is devoted in some form or another to agriculture, either to growing plants or growing livestock or growing plants that feed livestock. And in particular, livestock, just going up one trophic level in a food chain is extremely energy intensive. So it's estimated that a quarter of the world's greenhouse gases come just from raising cows for dairy and meat alone. And, that not only takes up land and resources, but it also takes up water. So something that often, or at least I didn't originally think about when I thought about the needs of growing food, are just the amount of water that you need to feed both animals as well as plants. And so, one of the ones that often comes to mind to me are things like, you picture the rice terraces here in the Philippines. One of the major things they're doing, right, that's kind of just so awesome and astonishing is just how they control water and how much of it is being sequestered there. And so a lot of what we have done on the planet to make sure that we have enough food to eat essentially reshapes the planet around us as well. And so I think increasingly when we think about the health of humans, we will also be thinking about the health of ecosystems as well. And these things go together. And now I think it's estimated that these costs are increasing almost in parity. And when it's been estimated by some that the costs to public health are now increasingly rivaled by the costs to the environment. When you think about transporting foods to where they're being sold, degradation to soils and ecosystems with the move to things like monoculture, and again, things like water use that are devoted to increasing industrial production of food, that poor diet, which, as I was showing before, has a cost on human mortality directly in terms of chronic disease, now is indirectly also having a cost on humans in terms of how we reshape the environment around us. So hopefully that provides at least, gets people thinking about kind of the importance of knowing what people eat and understanding what are the sort of forces that are driving global nutrition. And I think for me, where our lab is trying to contribute and help has to do now with how do we actually gather data on what people are eating. And this is something where it's really surprised me over the past couple of years how there still are open needs around innovation in this space. So there are a variety of ways that we track what people eat globally. But one of the most common and pretty much the dominant one when you ask what individuals eat around the world is you do this manually. You do it by asking them. So I'd say the vast majority still of epidemiological studies at the individual level occur by just asking people to fill out self-reports on what they've been eating over some period of time. It might be the prior day, it might be the prior month, or it might be the prior year. And I'm showing you an example of what it looks like for a very common dietary assessment tool that's used in the US called the Dietary Health Questionnaire, to give you a sense for hopefully some of the challenges associated with these methods. So you can see, here are the questions on the right. They're asking about the different, this is the pie section of the questionnaire. It's actually only a subset of the pie section. You see, we're on question 125 of this questionnaire, which we also are on subsection C through E of question 125. And it's asking individuals to recall how many different kinds of pies they've eaten. And they're breaking them down, as you can see, by cream, meringue, pumpkin, sweet potato, pecan. And then not only is it asking people to recall when they're eating, which of these pies they're eating, but it's asking them to recall their frequency over 12 months. And so if this already seems kind of mind-boggling to you, recall now too that this is page 33 of 40. And so someone is doing this, they're probably half an hour into this quiz right now. And like, this is how often still we gather data, really important data on what people eat globally. And so you can kind of imagine all sorts of challenges or reasons why this can be problematic, just to go through a couple. With these traditional modes of dietary data tracking, you can imagine that certain kinds of, often, people's emotions can get involved. You might not want to admit that you're eating certain kinds of foods, particularly if they're shameful or associated with poor health, like candy or chocolate. A big thing that hopefully you get a sense from this is that it's just very time consuming. If you've ever tried to track your own diet on a daily basis, you know that it's an effective means of weight loss because it just takes a really long time and it makes you very aware of what you're eating. Most people won't have the patience to do this, and I certainly have tried, and it's not really sustainable for long periods of time. It takes probably almost an hour sometimes to do these things properly. It requires high amounts of literacy and cognition. And so effectively these exclude large segments of the population from collection of diet. When we think about what people eat globally, if you're asking them to self-report, essentially what you're already doing is excluding what children eat because they can't fill these out. You're excluding the very old who might have cognitive decline and can't fill this out. You're excluding people who are undereducated, might not be literate. And then you're also excluding groups that might not be able to read in the language that you're administering the questionnaire. And then finally, a lot of this can be done for what you remember. You can't go way back in time, like 5 years back. Most people can barely remember. I can barely remember what I ate like 3 days ago. And also there are just all different kinds of versions of these things, and they're often hard to combine across many different kinds of studies. So what our lab does has been to be inspired by innovations actually outside of medical research. And in a way, we're inspired by environmental research, except it's a different kind of environmental biology. We're inspired by ecologists who studied animals in the African savanna. So Tyler Kretzenell and Rob Pringle, who are down here on the right, are two friends. And I was talking to Rob one day about how he studies these really adorable deer-looking animals on the left called dik-dik. And Rob was telling me these stories about how they do studies like the one shown here in the middle, where they look at zebras, elephants, impala, and they're able to track all of the individual plants, that's what's being shown here in the middle, that these herbivores are eating in Africa. And I remember being amazed, it's like, how do you get this much resolution on what these animals eat? You can't ask them, which is what we do for people. And he explained to me that they instead do something extremely clever with genomics. And it's something now that our lab back at Duke has replicated, and we're now working on bringing here to the Philippines, which is they are, or I guess now we have worked out that everything that we and animals eat on the planet, with the exception of salt and vitamins and minerals, everything else comes from living things, plants or other animals. And all living things have genomes in them. And the idea is that when we eat living things, their genomes, we consume them, they go into our intestinal tracts. They're digested, a lot of the DNA is digested, but actually millions of copies of food DNA survive digestion, and we poop them out. And so what we do is we collect that poop, which is easier than it sounds, and then we use advances in DNA sequencing to identify and amplify specific sequences of food genomes. We use DNA sequencing to read those sections of the genomes, and then we map them to reference databases that we have built of all the different plant and animal species that people eat around the world. So these reference databases on the right are a work in progress. I'll tell you more about the improvements we're making to this, but this is just sort of the general schema of what we do. So just to reiterate, because it'll come up a number of times here, the idea is that food has DNA inside of it. After we eat that food and digest it, that DNA passes through. And then we can use DNA sequencing applied to human waste to then reconstruct and identify what people have been eating from the residual DNA from the food they've been eating. And so we're really excited about this method, and so are epidemiologists that we work with, because it addresses a lot of the limitations that come up with traditional forms of dietary collection. Of course, it has its own limitations, and I could talk more about those later, but for now, I'll just point out that now what this does is it takes food tracking away and it makes it objective, meaning that you no longer have to ask people to rely on their memory. You don't have to ask as much time of them. You can directly get data from them. So for example, even from babies now, we can measure what babies have been eating, even if they can't speak, by collecting their stool. And then we can also collect data on what people are eating retrospectively. I was astonished to learn how many collections of stool there are that go back in the past, years, actually even decades in the past. And that can be accessed now to reconstruct what people have been eating previously. And this is now something also that is portable and translatable between cohorts. So we can look at what people are eating in Asia, in Africa, in the Americas, and we don't have to worry about differences of language. And we can always merge what we find because DNA is a universal language that maps across different kinds of plant and animal species. One of the big things I was telling you that our big efforts that we've been undertaking now as you can imagine, we need to have a body or a reference database of all the different genetic sequences of human foods. And so we've been building that. So on the right is a phylogenetic tree. It's a family tree of all the different plants that people eat. And one by one, we're slowly either taking publicly available sequences that have been previously sequenced, or, and this is sort of the fun part, going off and finding foods that don't have genetic sequences in public databases right now. And we sequence them ourselves, excuse me, and we add them to our database. So at this point, we have genetic sequences for over 800 plant species. This encompasses probably the vast majority of crops that are considered commercially important by the FAO. And but we're continuing to refine and add to this. And so you might be wondering, how accurate is this, or how confident are we that this has been working? And so one of the things that we've been doing to kind of show that accuracy is we've gone back to studies where we know what people are eating. And I happen to have done a study over a decade ago now, I guess, at this point, where participants ate foods from the plates on the left or plates on the right. This was a controlled feeding experiment that we carried out. And I should mention that we cooked the food on the right. Sometimes I realize this picture might not always seems misleading. It wasn't meant to see if what happens when you eat raw meat. It was more of just what happens when you eat vegetable-based diets versus ones that are animal-based. And what we find, each of these points now here is a different person in their stool, is when we sequence those stool samples and we ask what plant species are distinguishing stool samples from people when they're either on the plant-based version of our diet or the non-plant-based version, is the plant-based version. Those samples collected there are rich in things like oats, lentils, and squash, which is what we were feeding people. And so it's nice to see that it looks like the method indeed is working. So what we've recently published, and I can just share briefly here now too, has been an example of how we can use these sorts of data to start gathering data from what groups of people are eating or communities are eating, especially communities that we otherwise might have had a hard time gathering data on what they've been consuming. And in this case, it was a group of teenagers in North Carolina who came from households, many of whom were lower income and on government assistance. And because these were kids, especially, it was just hard to get dietary data from them. They weren't able to fill out long questionnaires. And it was also a cohort of kids with overweight or obesity. And because we were able to collect stool samples from these kids, we were able to use this genetic technique now to start reconstructing what they've been eating. And the plots here that I'm showing on the y-axis are a very basic statistic now about diet that we can gather, which is just counting the number of plant and animal species just the number of plant species that kids are eating. And we see patterns that I think are some of which make sense, others which are kind of surprising and fascinating. So for the ones that make sense, we can start here with income. So these are household incomes in North Carolina. And what happens is that as household incomes grow, kids have a wider diversity of plant species in their stool. So that seems to make sense. And it's also consistent with this plot here on the right, which is that for families who are food insecure, we see a lower diversity of plant species in their stool. So both of these seem to imply that for kids who have less resources, they have less access to a wider diversity of food. So things like that are sad, but also kind of expected. What's more surprising that we can now gather from these data is the following. One is that as kids get older, the diversity of things they eat goes down. So that seems counterintuitive. You know, my kids are right here on this plot. They're about 13 and 11 years old. I thought that they're supposed to eat more widely as they get older, but apparently what happens is as they get older, they have increasing agency as well. And it's people have observed that kids are less likely to eat sort of home-cooked meals, and instead they're more likely to eat out where they're probably not eating as well, or not as eating a high quality of food when they're eating out with their friends, say. And something else that we see is that kids with obesity actually eat a wider diversity of plants. And that's something else we'll return to later as to, I think that was a little surprising to us, and we're still trying to unpack. All right, but now, shifting gears, I want to share with you our group's work that's still unpublished. But I wanted to talk about it because I'm super passionate about it, and I think it's also relevant to the theme of sustainability today, which is basically thinking about global diets. One of the things that really attracts me now to studying global diet is just, when you think about diversity, it's harder to imagine something more diverse than human diet. It's estimated that there are hundreds of thousands of edible plant species. Only a fraction of those, 7,000, have been basically consumed by people with some regularity over the past 10,000 years. And even of those, there are at least about 100 plant species that are considered important enough and trafficked enough around the world to be tracked by the FAO. And these are considered commercially important. So there's a really wide diversity of things that people eat globally. But we actually often tend not to track that diversity for a variety of reasons. First is that, in part, there's a lot of regional variation, and it's basically just really tedious to gather data from lots of different parts of the world simultaneously. And then also many foods are actually informally eaten. So they might be foraged, they might be grown in home gardens, they might even be illegal to eat. So they might be basically being harvested out of places, oceans or forests, where people should not be harvesting them from, and therefore that they don't want to say that they're eating them. And other foods might not be getting tracked because they're not considered nutritionally important. So the ones that come to mind for me a lot actually are spices. So when you kind of think about it, spices are the most obvious thing to any human who eats. For what differentiates food around the world, it's the taste, right? It's not the calories per se that you pay attention to when you travel and you decide to eat something. It's how it's flavored. But often that goes unstudied, actually, because it's not considered to be a major driver of, say, carbohydrates, protein, or fat. And therefore, if it doesn't affect, say, obesity or stunting, often for global health purposes, there's less emphasis on it. And then finally, it's actually, kind of complicated just to think about the challenges with language and classification of different fruits and vegetables. Just how do you even put them into categories, right? So for example, if you go to the States, you think of a banana, there's basically only one banana, it's Cavendish. You come here to the Philippines, you think about banana, you go to the markets, right? There's at least half a dozen, you'll probably find a dozen different kinds of banana. How do you classify them? Are those all called banana and the same thing, or do you call them different things? These problems get, multiplied when you go across the globe. So we've been trying now to kind of wade into these questions on global diet and start seeing how genetic sequencing can assist us in sort of tracking at a wider scale, but with more resolution, what people have been eating around the world. And we've been doing this, members of our lab, Ben Nubert and Brianna Petrone have started this work, and we're doing it in collaboration with Laura Degas, and Amy Luke at Loyola University in Chicago. And we've been working with cohorts that Duke has been assembling in North Carolina and the US, as well as cohorts that Loyola University and Laura and Amy have put together in South Africa, Ghana, and Jamaica to study what people have been eating now using DNA sequencing. And so here is kind of a high-level summary of what we see. Each point on this figure is a different stool sample from an individual. So there are several hundreds individuals in this initial study that I'm sharing here. And the colors represent where those stool samples were collected. And this kind of plot is a kind of map. So you can imagine that each stool sample represents a vector of hundreds of different plant species that people are eating. And it's a count. How many times have you seen a plant species per person? It's really hard to make a plot in 100 dimensions. And so instead, what I've done is I've simplified that into just 2 dimensions, but I've done it in such a way that points here that are similar or close by to one another represent people who are eating similar plant species. And these arrows here are identifying why some people are plotted, say, on this part of the plot versus this part of the plot. People up in this direction have a lot more spinach, lettuce, cabbage, and carrot DNA in their stool, whereas people in this direction have more chocolate, corn, soybean, pepper, wheat, and potato DNA in their stool. And there are a couple of what I want to do for the rest of the time that I have is just talk through some of the patterns that we're seeing here, and what might be going on. So hopefully a couple of things are jumping out at you. Maybe the first that I'll point out is this first axis over here. This is getting at that question of diversity. Why is it that we're seeing higher plant diversity in people with obesity in our smaller cohort? We actually see that pattern here too. So folks who go off in this direction to the left over here, this is principal component 1. I'll be calling it PC1. People in this direction of PC1, more chocolate, corn, soybean, pepper, wheat, and potatoes, actually have a higher diversity of plant species in them. So the people who are over here, we see more plant species in their stool. And they also have a higher BMI. They often actually also have higher blood pressure. And so this is consistent with what we saw in our prior samples, or prior study. And so the question might be why or what's going on. And what is it about, like, aren't wheat and corn and soybeans, like, aren't these okay? Or these can be healthy. So this has been an excuse to start doing research with ChatGPT. Our lab has really enjoyed using it. And we've been using it to analyze USDA food data now. We've downloaded ingredient lists for over 7,000 different foods that are being tracked by the US Department of Agriculture. And they often look like the bottom over here. Scrambled eggs, sausage, hash browns, potatoes. And what we've been using ChatGPT to do is to take apart these ingredients now and decompose them into their component plant species. And then from there, we're trying to estimate why might certain plant species be associated with higher body mass index. And the reason is, I think that what we're finding is the following. So as I just showed you this plot on the left before, where the people who are on the left who have going this direction on principal component 1, they have more plant species in their stool. And this is what we see from the USDA data. It's on the right, which is that the more plant species we see in someone's stool, the more likely they are, the more likely a given food is considered to be highly processed. So sorry, I didn't say that in the clearest way. Let me try again. When we've analyzed our ingredient data from foods that are being tracked in US supermarkets, foods can be broken down by how processed they are. And so there's a semi-standard now measure of food processing. It goes from 1 to 4. 1 is unprocessed, 4 is the most processed or ultra-processed foods. And what we've been finding is that ultra-processed foods are much more likely to have multiple plant species in them. In fact, this has actually changed how I go food shopping. If I pick up a food and it has more than 3 plant species on it, I pretty much know it's an ultra-processed food and I probably shouldn't be eating it. And so what that tells us now is what's going on is we think that some people are having high plant diversity because they're actually eating a lot of processed food. And that's also why we're seeing this association with higher body mass index. And so, kind of for things that are just more consistent with that is, again, looking at our genetic data on the left. I showed you those arrows before. So PC1 was associated with corn, chocolate, wheat, potatoes, soybean, and pepper. And when we look at our USDA data, which is on the right over here, and we're asking, again, in USDA tracked foods, what plant species are most likely to be found in ultra-processed foods? The most likely ones to be found are things like sugarcane. We can't actually track sugar for reasons I can get into later by DNA sequencing, so we missed that one. But after that is corn, wheat, and soybeans. So again, it almost, another kind of rule of thumb now I'm using personally when I go food shopping is if I see corn, wheat, and soybean products together in a food, that's also a sign that it's probably highly processed. So a takeaway from this that I think has really interesting relevance to sustainability as well is higher plant diversity in in diets might not actually be a good thing, or at least some forms of diversity might not be helpful. And an example of this that I'm thinking of is this meal here, a Big Mac with fries and a coffee. If you count it up, has 19 different plant species in it, which were actually domesticated in probably 5 different continents. This is an incredibly globalized and diverse diet when you think about it. But of course, when you consider the environmental costs, there are both environmental costs to having this kind of diversity, and there's also health costs to this kind of diversity. And so basically what we're seeing from our genetic analysis is, at least I think, suggesting, in my mind, it makes me rethink what kinds of diversity in terms of dietary diversity we promote. It also tells me, again, like I was saying before, that when we see certain kinds of combinations of plants together in foods, that it might be a signature of food processing, especially, again, corn, wheat, potatoes, soybeans. And that often, certain kind of plant species, it might almost be a signal that these foods are heavily processed. And so just kind of as an example of this, to kind of show how this changes, again, like how I go food shopping, here's a food I'm about to show you on the right, and here are its ingredients. It's corn, wheat, soybeans, oats, rice, sunflower, sugarcane, and also there's DNA in there from honey. And so when you think about it, it's, at least to me, it's not obvious what kind of, I should say that based on the rules I'm suggesting on the right here, you might think twice about eating a food that has this many different kinds of plants that are being combined into it. But this is the ingredient list for actually something that's marketed as a very healthy, both from a human standpoint as well as an environmental standpoint, food. It's an organic cereal that's sold at a very expensive supermarket in the States. And so I showed this to kind of give you a sense for how thinking about now the genetic components of what we eat, specifically thinking about the species makeup of foods that we consume, I think kind of changes, in my mind at least, how I think about how we construct diets and what's both good for people as well as good for the environment. The other thing that I'll just mention here too, and I'll go a little bit faster too, because I want to save time for questions, is you might notice this pattern where there are only red samples up here associated with spinach, lettuce, and cabbage, whereas all the other samples from other countries are clustered down here, implying basically that these foods that I associate with, like leafy salads, are really only being eaten in the US, or primarily eaten in the US. And I'll say that, like, when we've expanded to look at other cohorts in other lower and middle income countries around the world, we see similar patterns. So we've included here samples from kids in Ghana and Nicaragua, as well as Kenya. Sorry, I actually had an updated plot with the Philippines in here too, but that result is just fresh off the press, and I didn't have a chance to incorporate it. But it's a similar pattern. It's for many of the countries, lower and middle income countries, they all kind of cluster over here. They're associated with higher intake of things like banana. Araceae are sweet potatoes. So these are basically starchy fruits and vegetables. And it's the US samples that tend to be living out more here by themselves with these salad greens. And that, to me, is also quite surprising for a number of reasons. And I've been thinking about that a lot. Why don't we see these as much, or why are they overrepresented in the States? Part of it probably has to do, or I suspect has to do with hygiene and sanitation. Often there are dietary recommendations, especially when people are getting away from urban centers, to be very careful about eating raw foods and to avoid foods essentially that can't be cooked or don't have a peel, which salad violates those rules. And also, when you think about it, these kinds of leafy greens are just per dollar, often calorically very poor. If you measure how many calories you get per dollar in things like lettuce and spinach, it's an order of magnitude below a banana or a sweet potato or potato. And those themselves are an order of magnitude below the number of calories that you get per dollar for something like flour, a more processed food. So I guess this shouldn't go to imply as well that processed food is necessarily bad. Certain kinds of processing have transformed nutrition around the world and allow much greater access to calories for people who have smaller budgets. Nevertheless, this might be explaining why these kinds of foods here are being overrepresented in wealthier countries like the US. So, just to kind of recap this or put a fine point on it, in my mind, it always makes me now think very differently when I hear someone talk about a Western diet, or that phrase comes up and you picture, when I ask people, what's a Western diet? They'll say, oh, it must be McDonald's, or it's a lot of bread and pizza. But actually now increasingly, thanks to things like globalization, if you go around the world, you could find McDonald's in probably every country now at this point, right? What our data are telling us is what's much still unevenly distributed globally are raw salads or foods or vegetables that can be eaten in a highly unprocessed and basically raw and intact form. And I think that it may in part be driven by culture, but I suspect a lot of that is actually not necessarily cultural, and instead it's socioeconomic or it's economic. It's being driven by things like sanitation and access to clean water, cold chains, and then as well as household incomes, the ability to afford a nourishing diet on a modest income. And so, tying back to the topic of today, which is sustainable eating, I'll just say that, this is sustainability now somewhere where we really want to head in a direction as we're collecting these kinds of data. As you can imagine, we're now building maps globally of processed food intake, as well as more intact and unprocessed food intake globally. And what we are looking for collaborators to do, so if there are people on this call who want to collaborate, please reach out afterwards. I'd be delighted to talk, is how do we tie the nutritional data and the dietary data we're gathering to data on things like environmental footprints, on the health of local ecosystems, the kind of how local food systems are being constructed. One of the questions we have is, do you see correlations between dietary diversity, the diversity of food systems and agriculture in like a local area, and then finally the environment locally? Do those things all go together, or do they enhance one another, or do they degrade one another if they're all, say, going down? And another place that we're heading on in this area to help us kind of gather data at these sorts of scale, I'll just mention, is through integrating with other data streams, like especially wastewater. I'll just mention very briefly. That's something we're excited about doing, is plugging into all of the wastewater collection infrastructure, which was started around COVID and is now being used for tracking other diseases. I don't have time to talk about it today, but we're actually using wastewater now to track the diets of entire communities or municipalities, even cities. Like, this can be done using genetic sequencing. And the hope is that this could all be put together one day and we can start having even richer data sets to help inform policymakers in terms of how do we guide people towards both healthy eating that's also more sustainable for the planet. That whole having been said, I was at an airport in Cebu over the weekend, and there was this quote that I'm showing on the left at a lechon store that like really stuck out at me, that I think also still shapes a lot of how I think about these things, which is that when people decide what to eat, myself included, right? You don't normally think about wastewater or directly. You don't think about like calories or sanitation. Maybe people do think about calories, but you definitely don't think about genetics or genomics, right? What people eat is driven by culture, habit, desire, craving, and then identity. I think that is extremely true. And obviously, policy and economics is only one component of how we kind of get people to healthier diets for ourselves and the planet. Also, too, just in case folks were curious or interested in collaborating, I'll say that some of the work that I'm doing in partnership with people at the Food and Nutrition Research Institute, as well as collaborators here at LaSalle, is working on building a genetic database of Filipino foods. As you can imagine, this is a real passion project of mine, particularly because it involves getting lots of food, taking samples, and then eating the rest. On the left here is a plot that of our very first pass of this that we've been putting together, where we we're looking at DNA that was collected from folks, I believe, who are in Marikina, and trying to identify the different kinds of foods that we can see in the Filipino diet. And we can see things like sitaw and singkamas, ampalaya, foods that are local to the Philippines. We see tilapia. But we also are missing quite a few things here. There's a lot of local fish that I don't see. I don't see bangos. And so one of the things that I'll be working on closely, or working on a lot for the coming months, is trying to fill in the gaps here and make sure that we have a resource here in the Philippines so that we can apply these kinds of cutting edge genomic techniques and use them to track the Filipino diet in its entirety. And I'm super excited to be collaborating again with Angie Lau, Jacus, Paul Yu, who's a graduate student here at LaSalle, who's moved on actually to Penn State, and Julia Gubat, who's at FNRI. Okay, with that, I'll close. Hopefully I've left some time for questions. I hope people are, please feel free to ask questions if you have any. I want to acknowledge our collaborators and sponsors who have enabled the work that I've been sharing with you. Extremely grateful to them, especially the ones that we were just saying at LaSalle and FNRI, as well as my lab back in the States, who's done obviously all of this work, and very grateful to work with all of them. Okay, I'm gonna stop talking, and then if there are, I can see some questions.
Okay, thank you so much, Dr. Lawrence, for your presentation. So may I invite our participants to give a round of applause to our speaker, Dr. Lawrence. Okay, we have a couple of questions in our chat box. First, this is coming from Susan Mendoza, and she mentions, good morning, Dr. David. Nowadays, Filipino loves to eat in fast foods. Do you think this contributes to increasing dialysis patients in our country?
So I'm not a nephrologist and an expert in dialysis, and all that having been said, I'd feel fairly confident with the conclusion that increased consumption of fast food is associated with things like seeing these increases in chronic disease that manifest in places like the kidneys. I will acknowledge that surprisingly, it's an open question, I think, as to why exactly, like mechanistically. We talk a lot about, for sure, fast food or junk food is not good for you, but it's surprisingly unclear exactly why. I mean, the associations are very strong. And I think things that I'll just speculate on is it might have to do with like taste, right? The food is just so delicious that you eat a lot more than you normally would. It could be due to like preservatives in the food or other chemicals. It could be that they're more digestible. And so that, if you think about eating something that has, say, a lot of fiber in it, you actually don't get most of those calories because you can't digest them and they're just excreted. Whereas things that are more refined, you absorb all of those calories. So the short answer is to your question, Dr. Mendoza, is yes, probably, but as to why, I think those are still important open questions.
All right. Let's move on to the next question. We have a question from Kevin Serona, and he mentioned, okay, let me just read. Professor David, have you looked at the relationship between gluten intolerance and Western diet?
Thank you for that question, Kevin. So no, not directly. And I'm glad you asked because this is, it gives me a chance to talk about some of the weaknesses of our method. So our method only measures DNA. It does not measure specific nutrients like gluten. And so therefore, it's tricky for us to directly measure gluten levels in people's diets. What we can do is look for foods that are enriched in gluten and then say that that plant species is present or it's not. But we're not always sure if gluten has been added or taken out. And so I'm glad to acknowledge at least some of the limitations of what we're doing. But it's obviously a very interesting and important question, especially for people with things like IBS.
Thank you for that answer, Dr. David. Another question from Samuel John Mareño. I think it's also linked from the previous question, but this is more
Yeah.
focused on oil. If we can detect various types of oil used in food processing, such as sunflower oil, canola oil, etc.
Yeah, this one, I think I'm a little bit more optimistic, and I think probably. So, for a couple of reasons. The first is, actually, I'll go back a couple of slides here. When we are measuring things here like soybeans in what I think are people who are eating processed food or more processed food. I don't think what's going on is that these people are eating a lot of edamame or tofu. I think what is one possible explanation is it's things like vegetable oil that's coming from soybeans. So I think, yes, it's possible that we're seeing it there. And then another example of where we might be picking out oil is here, when we're looking at how frequently we see different kinds of plant and animal species in the fecal DNA of Filipinos. Number 4 is coconut. And so it's possible that people are eating lots of cooking coconut and eating it that way. But I also think coconut is a common either ingredient as an oil or as a milk, right? Or a milk-based product. And so those kinds of semi-refined versions of coconut are still detectable, I think, in stool. How much is left are things we're still trying to figure out. I do think a lot of DNA is probably lost in the oil extraction process, but I do think that there probably still is some DNA left.
Okay, thank you, Dr. David. We have a question from Dr. Tan, more on the detecting the quantity of food. Can this methodology detect the quantity of food?
Yes, with some caveats. So yes, in that we can tell when people are eating more of a given food than not. So if you go off back to say these plots over here, someone who is over here does tend to have more banana in their DNA or in their stool than someone here. And so I do think that they're eating, say, more banana. What we cannot yet do, but I think could be possible one day, but it'll just require a lot of calibration, is we cannot say how many grams of banana they're eating right now. We lack that kind of ability to convert into those kinds of units. And the other thing I'll acknowledge that we are not yet quite able to do is to say relative intake of one species versus another. So it is hard to say, for example, that someone is eating more lettuce than potato. I can tell you that someone is eating more lettuce than another person, but I cannot tell you if they're eating within that person more lettuce than potato. And the reason for that is that the amount of DNA in a leaf of a plant is actually very different from the amount of DNA, say, that's in a tuber. It has to do just with the biology of plants. And so accounting for those relative differences is something we are also working on now. So that will come one day, but for now, we're still just looking kind of in a more relative fashion. It's a very good question.
Okay, we have another question, Dr. David, from Dr. Kathleen Aviso. So she mentions, how long does the DNA of the food we eat stay in the stool? Have you seen any shifts in dietary profile, for example?
Yeah, those are great questions. So going back to this first experiment we did over here, these are all the same people who over the course of 1 week are transitioning between diets. And so they're moving between the left and the right. So we've done some tracking experiments, and we think that DNA of food stays in our intestines for probably somewhere between 24 to 72 hours, and then it passes through. So I think of this as almost like a 1 to 3 day diary of what you've eaten. And then after a week, if you shifted your diet, you would see a change.
Okay. Thank you very much, Dr. David. Perhaps the last question. I have a curious question, Dr. David, especially when you shared that Subuchon slide.
Yeah.
Can you comment on, say, you have a graph on the income versus the type of food that intake that the participants have done? Can you comment on perhaps the cultural effect to it? Because some of the like regions in the Philippines, they would have a preferred food. Currently, I'm in Taiwan, so I can see that majority of the food here are more vegetable. Can you comment on that?
Yes. I can't, and my comment will be that I believe it's entirely true. And it's something that I very much look forward to studying in partnership with people at DLSU and FNRI over the coming year. That's also one of the areas that I very much want to research more. I'll say that we've been studying this back in the States, and you see a fascinating gradient across different parts of the States that have to do with geography. So if you go closer to the coast in North Carolina, you can actually tell how close a community is to the coast based on the different kinds of seafood they eat. So it's not even the total seafood, it's which species of fish are being eaten. Because closer to the coast, people are eating species that are maybe caught fresh, they don't ship very well, or they're not expensive enough to be worth shipping. And so I suspect at the very least in seafood, something very similar must happen here as well, and that you have different species of fish that are eaten regionally just due to availability. But I fully suspect that there's something similar happening in terms of where certain kinds of plants can be grown. And then perhaps the most interesting component of this, though, is what you're getting at, which is the cultural aspect, right? There's religion, there's like history, there's all of those kinds of ties, like the anthropology. People are wonderful in what I think what we choose to eat and how we assemble our dishes, right, is just another manifestation of human culture. And so that's exactly the kind of place that I would love to kind of observe and gather data on. And I don't think that we could always necessarily with policy change these things, nor should we, right?
So, Dr. David, just a follow-up question from Samuel John Pareño, and he is asking if we were to eat cow, is the method able to detect the diet of the cow?
Those are good questions. Probably not, unless you are eating the intestine, which happens, right? And so, but I'm glad you also bring up cow because that brings up another limitation of our method, which I should acknowledge, which is that at least with this method, there are other ways to do this, but we cannot tell the difference directly between milk and beef. They have the same DNA, right? And so there are other, techniques that we're developing to tell those apart. But that also just reminds me too, I think I believe it's like avocado and bay leaves actually come from very similar kinds of plants. And so when we see that kind of DNA, where you can't always, again, be certain what people are eating. So that's just another limitation I should acknowledge.
Okay. Perhaps one more question, Dr. David from Dr. Tam. Dr. Tan mentioned, have there been attempts to map correlations between food DNA and gut microbiome DNA?
Yeah, that's a great question. And yes, we have made a lot of those attempts. That's actually why we started this research, because historically our lab has been a microbiome lab. And so we gathered the food DNA so that we could answer questions about the microbiome DNA. And honestly, one of the most surprising things here to me is the answer has been usually no. Those associations are quite weak. And there are some exceptions. So if people are very sick and say they're like in certain kinds of cancer patients who have been getting a lot of antibiotic treatment, they're very sick. And so their microbiome is also distorted. Sometimes there we see an association. And also in babies, we see an association, which I think is fascinating. And maybe that has to do with an association between how people, specifically when you're introducing solid foods and withdrawing breast milk, we can see when weaning is occurring, and that is being associated with microbiome development. But for the vast majority of other people in sort of normal life, we see less. And maybe that's something I would love to talk about at greater length maybe one day, because why that might be the case. There are a myriad of reasons why it could be.
Okay, I think that's all the time that we have for the Q&A portion. So again, thank you so much, Dr. Lawrence David, for sharing your expertise and answering
Yeah.
all our questions. So let's give Dr. David a round of applause.
Oh yeah, thank you so much for inviting me to speak. It was a joy, and I hope to get to meet all of you in person soon.
All right, so thank you so much. So that actually concludes our lecture for today. So at this point, may I just acknowledge the presence of Brother Joe Scheiter. Good evening, brother, or good morning.
Good evening here. Good morning there.
All right, so let me just share my screen. I'm sharing here the link to the feedback for us to further improve our delivery of the LaSalle Sustainability Lecture Series. So we would appreciate any feedback for this session, and we invite you for another lecture series by next month. And we would just post the details in the social media account. So again, thank you so much. I'm again Dr. Aris Obando signing off for this lecture series.
Thank
Thank
you
you,
all.
everyone. Thank you, Dr. David.
Yeah.
Thank you.
Oh.